Hello, everyone. I'm Jeremiah Jesse Lance, and I'm here to talk to you about understanding biological models in code. Many of you have probably seen me talk before, either at an earlier DevCon or just in general. And to those people, you've probably already tempered your expectations, which is really good. To the rest of you, and to the poor souls who subject themselves to this recording later, it's probably good to level set what this means. Understanding biological models with code, understanding biological models just as a concept is a really lofty goal. There are people who spend decades doing this for a small portion of biological models, and this is not a higher education course. This is a 40-minute DevCon presentation. So I need the understanding from you, participants, that the information that I'll present on biological models will necessarily be incomplete. You're not going to come out of here with a degree in biology. Hopefully you didn't think you were going to. What this presentation is a personal journey that was revelatory for me. I learned some things from it, and I was hoping I could share that with you and provide some value. So let's move on. Why? Is this thing going to work? It worked earlier. There we go. So, other than just the fact that I'm a curious individual, this is a topic that interests me, and hopefully you, and that's why you're here, I had personal reasons that started me down this journey. And the first of those reasons was really the musculatory system. And being the data-oriented obsessive nerd that I am, it wasn't really muscles that interested me, it was optimizing muscle gain. That was what interested me, and that's what I wanted to do, being a fan of game theory and being a little bit obsessive. So in going through this journey and learning everything I could obsessively about muscles, I got to the point where I needed to know what sources of truth were. And all too often, the sources of truth look like this guy. And it's the big guy in the gym who's going to teach you that if you work out two different parts of your body on the same day, the protein's going to get confused and it won't know where to go. And he'll be real confidently wrong. And that's kind of the first source of truth that many people who go down this journey meet. The YouTubers, this is Dom Mazzetti. But that's not what jives with me. That's not what I'm looking for. That's not what resonates as truth. Eventually what did resonate as truth was this. This is my source of truth for understanding muscular models. It's an empirical study that has citations. It goes through all of the ways that they've introduced this and comes out with a result. And this did resonate with me. However, as an engineer, it wasn't really how I'm used to assimilating data about models. And the concept of making an empirical study about, say, the Google Map API, where instead of looking at the documentation, you go through and say, well, I attempted these many requests with these parameters under these scenarios, and this was the result I got, and that's how Google Maps API was documented. It's ridiculous. So I kind of wound up with a hybrid approach of assimilating this data. I would go through the studies, but in my mind, I'm really kind of diagramming, right? I'm coming up with a diagram that makes sense to me as an engineer. I've turned it into a system that is formulaic and makes sense. And I kind of knew this was naive at the time, but this is how I assimilated the data, and this is how I kind of understood how the system worked. And honestly, it mostly worked good enough for me. I was successful in building muscle, believe it or not. Eventually, my naive understanding kind of became a problem. I came into a system that I thought I understood well, and something was going wrong. And it was because I had personal medical problems, which aren't really the focus of the talk. We don't need to go into those. But essentially, I thought I understood a model, and it was becoming obvious that I didn't. Something wasn't working the way that I thought it should, and I was not being successful in understanding it. And this was kind of when I came to the revelation that perhaps the way I was thinking of biological models in general wasn't only naive, it was just fundamentally wrong. So how could I correct that? And what I decided to do was create biological models in code so that I could understand them better, because I'm very good at assimilating data about code. I'm very good about understanding systems, I think. And I was hoping that this exercise would help me understand the biological models better as well. So it made sense if I was going to start at modeling a biological model, I would kind of start at the beginning. Start at the most simple form of life, meaning the first form of life that began to exist on Earth. So in the beginning, this is about 4 billion years ago, life started to take a turn. There was an epoch around 4 billion years ago. And of course, before 4 billion years ago, this was still happening. Things that we would identify as life existed. The difference was they just started dying. So they would exist, and then they would die. And it didn't really matter that they existed. This happens all the time. It still happens. What changed was 4 billion years ago, there was an epoch where these things that began to live began living better than they began dying. So it kind of tipped the balance. And the first form of life that we recognized was prokaryotes at this time. And prokaryotes are single-celled organisms, and they essentially absorb things via osmosis. How they've defeated this time problem of coming into existence and then dying and not mattering was that they reproduce, and they reproduce via binary fission, or sometimes biological fission. And it kind of looks like this. Essentially, as they osmose into things, they split and they become duplicates of themselves. So that way, when they die later, inevitably, there's still more of them, so they continue. And that's kind of the epoch that happened 4 billion years ago with prokaryotes. But essentially, they live, they absorb things, they die. Pretty simple model. And this is what they look like. Some examples of prokaryotes at this point were bacteria and archaea. It's not really important, the difference. We'll just wave hands and say, I told you this would be incomplete. All right, so prokaryotes and code. The language I chose to model this, because I knew I'd be representing cultures, is Golang. And the language doesn't really matter. It's really good at asynchronous processing. I do have a blog post that actually goes into all of the objective reasons why Golang is an optimal language, but that's a different thing. You can look it up in Connect to UCERN. But one of the rules for coding this that I wanted to strictly adhere to was that since these entities don't really have a system of API calling other cells, a strict rule I wanted to follow was no cell would have a pointer to another cell, and nothing would have a pointer to a cell. So essentially, they live in this environment where they can't point at any other address in memory. So let's look at structure, what I came up with. So again, hopefully the syntax isn't particularly important, but we have a structure of a cell. We have an osmosis stream. This is a channel that represents proteins coming from the outside world and interacting with my cell. We have an observation stream, and this is actually just something I needed so that I could provide a demo for you guys later. It doesn't actually have a concept of an observation stream, but I needed some way to represent what was visually happening, either emitting photons, emitting waves of sound, some way that you could see something in a log or something. And this was my compromise of not having a pointer to anything, but there is a stream that I could send events to that indicate something happened. I have a hunger integer, and this hunger is kind of an anthropomorphization, meaning that I've treated it as human. It's not really hunger, it's really how satiated the cell is, but basically it's, am I starving or am I full? There's a channel that indicates whether it's died or not. The reason for that we'll get into later. And then there's identity and identity chains. So really, of the 6 fields that I've put in this structure and this global, only 3 of them, the death channel, the hunger integer, and the osmosis stream, are actually relevant to the concept of representing prokaryotes. The astute viewer may have noticed I didn't mention a geographical location of the cell, and this is honestly because geography doesn't wind up mattering. We actually model a lot of things, even computer science modeling geography is relevant in, but we never model it. And an example is, say we have our source code, and we're like, oh, well, here's our source code. Well, that source code isn't what you're actually creating. What you're actually creating is machine code, right? And you're like, well, no, it's not actually machine code either. It's binary. It's actually binary. But it's not even binary either. What it actually is a state of ohms going through transistors in a specific geographic array. So when we're writing code, we're actually writing something where geography matters. The position of this does matter. However, we don't care about it. We don't model it. We don't care. So I'm not modeling it either. We're instead using the clock as a randomization element to determine when a protein collides. So constructor. Yeah, sorry for that detour. I had to throw that in there because I'm pedantic. Start at the top. So this is called a constructor. Honestly, there's really only this 1 second line that actually creates an object. But let's see what we're being called with. It's being called with a channel of proteins, which is our osmosis stream, some channel of observation events, and some identity chain. So we're not getting other values that we might not expect to be getting. Then what this is doing, time.tick creates a channel which receives an event every unit of time. In this case, 1 second. So every 1 second, time.tick receives an item. It's an empty interface. It doesn't matter what it is. Essentially, this is a repeating tick of time. So then we create the cell. We pass in our osmosis and observation stream. We make a channel of Boolean, which will be our death stream. We pull something off the identity to determine who we are, and then we pass the identity chain itself. The reason being that as these things reproduce, we want to make sure things that are happening asynchronously don't wind up with the same identity. That's why I have an identity chain. In a massively asynchronous system, I don't know of another way to do this. Someone could probably tell me later. What's important about this, though, is that we create a thread. In Go, when you have Go and then followed by a function, it spins off a thread to execute that statement, and then does it on a different thread, and then continues through here. So essentially, I've created an honest method and called it with my cell, and then this cell is now the only pointer, or this loop is now the only pointer to my cell. So basically, we're selecting either I die or I get a tick element, and it'll wait there until one of those happens. So every second, it'll be calling this until it dies. In that case, it breaks it, everything gets garbage collected, and my cell's gone. So we need to know what tick does. So the cell's tick method also is doing a select off of these channels. And it's essentially looking at this protein stream, again, or the osmosis stream. We should probably hop down to the default. It doesn't wait for this because it has a default, and because it has a default, if that means there wasn't a protein, a second elapsed, I tried to eat, there wasn't anything there. And we kind of go through the hunger stream. We get a little bit hungrier, maybe we starve, and if we're past starvation, we die. So pretty simple there. If there is a protein in the osmosis stream, we osmosis. So we'll look at the osmosis method in a little bit. But here's kind of the interesting thing. If we weren't hungry and we just ate, and then there's more food and we're continuing to eat, we'll split. And that's kind of the prokaryotes binary fission. That's how we're representing it here. And here's some other things that had to exist for the cell model to work. Osmosis, which all it does is say, I ate something, and then it calls react. React, which just kind of takes 200 milliseconds. And then split, which just calls a new cell. And note it doesn't pass a pointer to itself, it doesn't keep a pointer, so I've kind of kept myself honest on not sharing pointers between these cells. All right, but these cells aren't really exciting to operate on. We kind of need an environment for them to live in so that I can model them. To do that, we're kind of missing the top of this. This is an array of proteins. In our case, protein is an empty interface, so I've chosen to represent it as empty string. So what this is a representation in time from 1 second to 31 seconds, where I'm passing varying groups of proteins. And the reason I did it in this particular shape, I did it so that it was kind of graphically pleasing to you, but essentially every second it'll go up or down or up or down. I was attempting to model waves at this point, an aquatic region where I assumed that it was happening. It actually turns out, after I got into studying this, that it's equally likely that the earliest prokaryotes were in igneous rock as opposed to water, but I was trying to model the waves. That's what I was doing here. Sometimes there's a lot, sometimes there's little. It could really be any cycle. Since we're talking about 4 billion years, it could be a cycle of eons. It could be a cycle of millennia. Any cycle. And then to actually kick it off, to actually go down the route of abiogenesis, we have an environment that just makes a stream of proteins. That's what it winds up passing into that very first cell it creates. We do our own time of ticking here and create our own cell. And then we loop through that environment array I showed you earlier. All of those proteins eventually just get fed in array grouped into the system every second on a tick. And here is what that looks like. Now, I created an observation module just for this, just to show you guys what this looked like graphically. I learned how OpenGL works in Go. All right, so here, we got some cells. This is around when the protein starts spiking up. We got a lot of them. And the protein kind of goes down, we die off, we got homeostasis. These are gophers, by the way. I call them Eukaryotes. We got another spike, so again, they reproduce. Again, the protein stream goes off. They consume the available food and start dying off until eventually the last several ticks of there had no protein coming into the system, so of course they all starve off and die. We can also visualize this via chart, and it kind of looks like you'd expect. There's spikes of protein, there's spikes of Eukaryotes that represent that, and it just kind of looks like hopefully what you would expect it to look like. So we did it. We modeled prokaryotes, but now to assimilate this and to have it help me in any productive way, I need to compare it to a system that I understand. I need to be able to say, what does this system represent that I also understand so that I get a better understanding of what's going on in this biological model specifically. And it actually took me a while to find a system that behaved in this way of prokaryotes, but I did eventually find one. This is actually a picture of requests per second for a self-scaling Amazon Web Service node. And you'll see here, it's monitoring requests per second as this green line over here, scaled, looks like 2,000 per line. And it goes per hosts over here, scale on the right side, looks like about 12 per line. So as the traffic spikes up, we get a lot of hosts. As it dies off, the hosts go away. So this actually really closely models. It even has a cycle. It's a cycle of days. Here, let me get a drink. Apologies. So essentially, that really closely behaves. There's a difference of scale. Obviously, I'm scaled at more like 2 to 1, but they're scaled at more like 1,000 to 1. But essentially, if I'd rewritten my chart like this, it would look kind of like you'd expect a self-scaling web service chart to look. You'd see requests spike up, you'd see your nodes spike up, and so forth. So that was kind of neat. So what did I conclude from this? What did I learn from this first biological model? First, it was actually kind of shocking that a model that was biological in nature so closely mapped to a computer system model in an unintentional way. I didn't intend for it to map to Amazon Web Services or scaling web services. That wasn't something I intended, but it certainly did. Like it maps very much the same way. And the second thing that I learned from this was that in the model as a computer system, the prokaryote doesn't really represent like a concept or a piece of functionality. The prokaryote was really representing a computer. Like it really was a host in this equivalent model that I found. All right. So we completed that, but I'm not one who's going to rest on his yannies. That was a Laurel Yanny joke. Yeah. Won't rest on my laurels. So the next biological model. I wanted to make sure that we could continue down this stream. And of course, the next thing that would make sense would be the next form of life. We'd go forward in time. So at this time, we have a bunch of prokaryotes. They're relatively successful. And over the next 2 billion years, because they're not really a homogeneous group, they become kind of interdependent. So there are some prokaryotes that consume protein A and they emit protein B. There are others that consume protein B, they emit protein C. And this kind of wound up leading to a symbiotic relationship between these cells. These prokaryotes wound up depending on each other. And as that happened, over time, they kind of made this formal. Essentially, they were already symbiotic, so they kind of became eukaryotes. Now, eukaryotes are different from prokaryotes in a number of ways. The most strict speaking is that they have a strict nucleus, an actual hard shell that indicates something's outside of me, something's inside of me. They're not just absorbing anything willy-nilly. The other differentiation is that they have organelles. This is another one of those hand-waving, incomplete things. There are prokaryotes that have organelles as well. We're not going to get into it. We're going to pretend as though the strict nucleus and the organelles are the difference between eukaryotes and prokaryotes. For reference, all human cells are eukaryotic, with the partial exception of red blood cells that begin their life as eukaryotes and then shed their organelles. They just freaking toss them out of there. It's probably also a college course on red blood cells, so don't need to get into that. So the code model. How does it change from prokaryotes? The primary difference in the structure is that we've added a nucleus stream. So now, how we represent being geographically outside of the world is that you interact with the cell via the osmosis stream. You interact with the eukaryote and you get osmosis in through that nucleus. But there is a representation of the inside, which now is diverse from the outside world. There are still organelles in there that still need to be fed. They still absorb in the exact same way. They still essentially are prokaryotes, but they can no longer get from the outside world. So instead, there's a representation of being inside the cell. So the nucleus stream is inside the nucleus of the cell. Otherwise, the structure is the same. For the constructor, all that has really changed is during this creation, because we are biologically fissioning out and we need a certain number of organelles simply to survive, there may be biological fission that happens and the cell simply dies, but that's not what we're worried about. We're worried about modeling successful binary fission. We create some organelles, and this would be just encoded in the RNA. It'd be whatever happened to be part of this eukaryote. It would split and succeed and have these new organelles as well. And then, of course, in osmosis of the eukaryote, we send some different proteins into our interior stream. So basically, we pulled it in, we've done some processing, we sent some different proteins into our interior nucleus stream. So now, we can chart this out. And as you might suspect, it looks pretty much exactly the same as the prokaryote model. And at this point in my journey, as probably many of you in the audience are also feeling, I began to wonder if my time was being wasted. But we carried on. So we have this chart. We represent our eukaryotes. But we really know inside of that, there's that other chart, the chart we saw earlier, which there's prokaryotes inside. It behaves exactly the same. So we've kind of inceptioned here. We have eukaryotes that pull things in. They give it to their stream of organelles internally, and they look like this. So what conclusions did I draw from this? The primary conclusion was that, honestly, while it's a huge step forward in the variance of life, it wasn't really a huge step forward in the model. Explaining it's basically just as easy. Like you say, oh, it's a cell and it has some organelles. It didn't really get more complex. However, the modeling complexity became multiplicatively larger. We're no longer representing one thing when we say we have a thread. We're spinning off threads for several things every time we had a thing before. So essentially, with 2 organelles, we've multiplied our number of threads by 3. It's just computationally more expensive. There's a lot more going on, even though the model as a concept hasn't really changed. All right. The final model. So we move forward in time again, because I wanted to go to something that was a bit more complicated, but linearly so. And we eventually get to a point where there are types of eukaryotes. And these eukaryotes are also not a homogeneous group. They become specialized in certain ways. And some of them develop such that they grow in colonies. Essentially, they find very similar eukaryotes. They live together. They function in the same way. They geographically are present together. And they function basically as a colony or a unit. And this eventually becomes the first land fungus. Now, land fungus was eventually a unicellular organism of eukaryotes that just was classified as fungus. It eventually became a colony, though the original fossil, the early fossil record of fungus is actually really poor. So I won't say nobody knows, but at least I don't know what the original land fungus looked like. But what's also equally sparse as the fossil record is the stock photos in the public domain of fungus. So we know they don't look like this, but any venturing mycologists who want to go out and take some pictures of more primal fungus could probably help out the world. So fungus as we're speaking here, is when it became a unit. These colonies no longer just were individual units that just happened to live near each other. They began to form structures such that they were acting as a unit. They became insular to themselves. They kind of had an outside of the fungus world and an inside of the fungus world. And you'll see where I'm modeling this. I kind of had a premonition where this was going when I started this on my journey. What does the fungus model look like? And I was like, well, I've got the outside of my fungus in this osmosis stream, and then I've got what's inside my fungus on this fungus stream. Like, wait a minute, that sounds really familiar. Like, wait. Yeah, that's exactly the same as a eukaryote. What's going on? So, we model it, right? We chart it out. I'm like, wait a minute. I know it looks like this. I know inside of it, after I've pulled things into my fungus, there's eukaryotes who pull from a stream. And then inside of that, they pull inside of themselves. And then inside of their nucleus, there's prokaryotes. Whoa. So basically, let's talk about some conclusions. I learned that biology is really good at inheritance. The same model actually kind of repeats itself. We'll do some more hand waving here and jump to the human body. The human body essentially works at the same realm. We have a gastronomic stream where we pull in proteins. We have a respiratory stream from the outside. We pull them into ourselves, and then we have entities at that point which pull from those streams, say our lungs, our heart, and they in turn feed to other streams, our bloodstream, our limbic stream, our central nervous stream. And the organs as units kind of work at this level too. Like even something complicated like, say, the pancreas, it's pulling from the bloodstream, but then internally it has islets of Langerhans in it which do certain things. So really there's a number of channels inside of the pancreas, even as just a small portion of the human body. And it turns out it doesn't really stop there. It turns out it's still relevant for macrobiology too. Macrobiology meaning groups of living entities. This is what's called a Lotka-Volterra equation, and I apologize if I mispronounced that. I've only ever read it. I've never heard the word spoken. But a Lotka-Volterra equation, getting out of here, it was an equation used to describe predator and prey interactions. And predator and prey is kind of misnomer. It could be vegetation as it comes in and the herbivores that eat them. Essentially, a supply and demand of food to recreation or reproduction and how it achieves homeostasis. And it turns out that this is even applicable to humans. Lotka-Volterra equations have applications in economics as well, which is essentially the group dynamics of humans in a supply and demand system. And it functions essentially the same. Something comes in, it gets processed, it goes out. So I wanted to evaluate, has this helped me understand biological models? Have I learned anything from this process? And the first thing that kind of hit me was, well, duh. Of course this thing's universally applicable. Basically what I've modeled is take an input, do a process, and then do an output. Like, it totally makes sense that would be universally applicable. I haven't really made anything revelatory, and I know you're again thinking about the wasting of time, but hopefully we'll get you something of value. So in terms of achieving my goal of trying to understand a biological model, maybe I did some success in learning some of the specifics early evolution, but in terms of understanding biology as a whole better, I think I failed. I didn't do that. What I did do, I think, was understand why I don't understand biology better than I do. And really the thing is, I was thinking about these models as an insular system. And really what it's more akin to is understanding how the internet works. These entities aren't working as a system. They're not working in some particular diagram or design. They all have their own, I'd say intent. They don't really have their own intent, but they all have their own replicatory life cycle. None of them are actually doing anything at a grand scale. They're all doing an input, process, output. They're just doing it at super huge scale. And it turns out, as I was looking through this, it collided with another field that I really liked looking into, which is cognition, and specifically cognitive bias. Since I was so bad at understanding I was bad at reasoning about biological models, I wanted to know why. And it turns out there's actually a cognitive bias that addresses this. And this is a variation of reasoning via analogy, and it's anthropocentric reasoning, essentially reasoning about something as though it is a human. And you may think back to our original model, where I had hunger as a prokaryote. That was already an anthropomorphization. It wasn't hungry. I just chose that because I'm used to reasoning about things as though they're human. And it was amusing to me when I realized that even when I'm reasoning about the human body, literally reasoning about an anthropoid, using anthropocentric reasoning was fallacious. It was still wrong. But where it isn't wrong is when you're reasoning about a system a human has built. The reason we're good at reasoning about systems is that this bias, this precondition where we assume something is like a human, is actually perfectly viable when we're reasoning about a system a human built. We think, well, how would I have built this? How should this function? And it turns out that that line of reasoning perfectly aligns to how a human would have built it. So really, the bias works for us in the computer science world, and it works against us in the biological world. And that was kind of how I learned why I was bad at biology. However, I think there's more to it. In the past couple of years, the world's best Go player, not Go the language, but Go the chess-like board game, has been a system that was not programmed by humans. Chess, for the last more than a decade, the best players of chess have been systems that were not programmed by humans. It's reasonable to expect in the next couple of years the best drivers in the world will be piloted by a system that was not designed by humans. So while this bias is perfectly reasonable now, when we use them to reason about the systems of the future, the systems that are right around the corner, it may not be. Like, we'll be reasoning about a system as though a human designed it, and it'll be created through machine learning. Machine learning's, I think everyone can agree that's the future. And it's just, I think, important to keep in mind that if we have this bias, we should know about it so that when we are reasoning about the systems of the future, we understand how we might be reasoning about them incorrectly. So that is where I will land. We have this slide so you can check in and give me feedback. Thank you very much for sitting here and listening to me. I'll be up here. I'll be ready to take questions if anyone has any. But otherwise, thank you.